And now, let's deep dive into life science and AI partnership. It is not surprising that these fields are merging more and more over the last decade. I would like to give the stage to Jérôme Tubiana, postdoctoral fellow at Tel Aviv University, who will talk about the interpretable prediction of protein binding sites with geometric deep learning. Jérôme is a FSP postdoctoral fellow at the Tel Aviv University, collaborating with Professor Chaim Wolfson and Dina Schneidman. He is developing AI tools based on geometric deep learning for better predicting how proteins interact with one another. Previously, Jérôme received his PhD in statistical physics from École Normale Supérieure de Paris. Under the supervision of Professor Rémi Monasson, he developed novel physics-inspired, data-driven models of interacting biological systems. Let's invite on our virtual stage Dr. Jérôme Tubiana. Good day to you. Do you hear me well?
Yes, very well. Thank you.
I'm sorry if I mispronounced any of very difficult things for me to pronounce, but I do believe you will allow us to deep dive in something very difficult in a very easy way, right?
I hope so.
Okay, let's discuss this after your presentation, and now the floor is yours.
Thank you very much. Thank you very much to the organizers for the invitation. I'm quite excited to share a few words about the research topic in general and our research in particular. So today we'll talk about proteins and how do protein binds, and how can machine learning help us to better understand this phenomenon. So a couple of words first about proteins. Of course, you hopefully all know about what are proteins. These are biomolecules that are encoded into our DNA and basically perform all the functions in our body. They can transport molecules. So let's say hemoglobin is a protein that can transport oxygen from our lungs into our limbs. They can catalyze reactions. So for instance, trypsin is a protein that digests elements and helps us turn them into energy. They can be sensory. So photopsin will be like, if you look at how our eyes work, it's yet another protein that is sensitive at light and help us detect, actually see. Proteins actually help us defending ourselves against pathogens. Antibodies are like our first line of defense against, let's say, viruses like the coronaviruses. They structure our body, they also regulate our activity. So regulation is, for instance, making sure that the genes that are involved into the development of our body once we are young are not active anymore once we reach adulthood. So a couple of words about what are proteins in terms of chemistry. Protein is a sequence of amino acid. An amino acid is a small molecule that can polymerize, meaning that they can bind to one another and form a very long chain, which we call a sequence. So there are 20 types of amino acids, and if we let's say want to construct a protein of size 50, then there are 20 to the power 50 possible combinations of amino acids that construct a protein. So proteins are objects constituted of simple ingredients, proteins, but amino acids, sorry, but they are essentially an infinite number, and every protein is different from one another due to its sequence. These proteins, once produced by our body, they will adopt what we call a structure. This molecule will fold onto itself to adapt into a shape that we call the protein structure. And this structure provides it with its function. So for instance, this protein will function is to recognize, bind a certain molecule, and the binding will be deeply mediated by the structure of the protein. The arrangement of the amino acids into a specific conformation will make the binding possible. So one very important aspect of proteins is that they do not work alone. Proteins interact with one another. So for instance, actin filaments in our body, they would polymerize into longer chains, and this is what provides us with structure in our cells and in our muscles. You can have very large complexes of proteins that would sort of self-assemble, constituted by hundreds of proteins, such as this nucleopore complex, and together they would form macromolecular shape that, let's say, serves as a communication channel basically between the nucleus and the cytoplasm of a cell. Sometimes proteins interact with one another so as to regulate themselves. This is here on the right, you can see an example of a so-called enzyme inhibitor complex. So this is an interaction between two proteins whose goal is to prevent, let's say, the orange proteins from functioning. You do not want trypsin proteins that digest other proteins to be working around and digesting proteins of your own cells. So this is where these inhibitor proteins come very handy in our body. Sometimes these proteins can would collaborate with one another so as to signal, transfer information from stimulus, let's say light, into producing a complex response, let's say feeling awake like during daytime and feeling sleepy during sleep. This is achieved using these so-called biological gene pathways that can adapt the behavior of a cell to its environment and its stimuli. Finally, in immunity, you can have interactions which are whose purpose is to bind and neutralize a pathogen. So this is an example of complex between an antibody, in red and green from our body, and the so-called receptor binding domain of the spike protein of the coronavirus. So this blue protein is the protein that is responsible for the entry of coronaviruses into our cells and in turn infection. So proteins are everywhere in our body and in all biological organisms. And what we would like to do is to better understand how they interact and also to predict it quantitatively. We would like to know, if I take two proteins, whether they will bind to one another. We would like to know how strong the binding will be. And we would like to know what will be exactly the shape of the binding. So there are many applications, but among them, here are a few. You would like, for instance, perhaps to inhibit a certain protein, which is overactive during disease, let's say cancer. So our goal would be to design a protein like the green one, an inhibitor that can bind to, let's say, this enzyme and prevent it from being overreactive. In the context of immunity, for instance, it can be quite different. We would like to know, suppose that we have a set of antibodies that we isolated from the blood of a recovered coronavirus patient. What we would like to know is whether these antibodies will work against future variants of the coronavirus. So we would like to know if this given antibody will bind not only to this so-called receptor binding domain of SARS-CoV-2, but will it work against similar proteins that feature mutations that can potentially alter the binding. So could we sort of come up with antibodies that would work for, let's say, SARS-CoV-3 or other related viruses? So and finally, like generically, you could think of doing protein design, which is designing de novo protein for your own purposes. Let's say, could we design like sort of artificial actin filaments that we could use for robots, for instance? Okay, so just a couple of thoughts about the physics of proteins. So how are these biological processes mediated? How do they occur? It's actually physics. For instance, amino acids they have 20 different types of amino acids, and each of them have different physicochemical properties. For instance, some of them are electrically charged, some are not, some are polar, some are so-called hydrophobic. They sort of like tend to avoid water, and they prefer to aggregate within themselves. And really, it's these physicochemical properties that drive the behavior of a protein. So, for instance, two charged amino acids with the same charge will tend to repel each other due to the Coulomb electrostatic force. So you can, in principle, integrate all these forces into a physics model, which is called and solve the so-called equations of motion. Here, this would be the Langevin equation. And in principle, everything we should know about a protein is encoded in this equation. I'm going to show you here a short simulation of folding a protein. So as you can see, the sequence starts completely unfolded just as right after it was produced by organisms, and will just by the sheer the forces that with which amino acids interact will drive the protein towards a certain shape. So the problems with these simulations is that they are extremely computationally intensive. This short simulation, which is not doesn't even show a complete protein folding process, took a very long time. I think it required a supercomputer in this case. So what we are looking for are heuristics that are as accurate, if not more accurate than our physical models, and that are much faster. So this is where sort of computer science gets into the protein world, and I will give you a quick overview of how people have conceived algorithms in this domain. So, for instance, a simplification of the binding process between two proteins is the so-called lock and key model of biomolecular interactions. The idea is very basic. It's simply that stating that two proteins, it's an enzyme and a substrate, have a certain shape complementarity. Basically, the substrate would be convex, where the enzyme will be concave. And this is this shape complementarity together also with physicochemical complementarity. Think about, let's say, opposite charged that are near one another, so that there can be an attraction between the two proteins that will drive the binding. So very much alike a key and a lock. Shape complementarity drives the binding. So this idea has motivated the development of so-called heuristic algorithms like rigid body docking. So suppose that you know the structure for two proteins and you would like to generate a model for the complex. Well, you're going to try to analyze the shape of these proteins, look for regions that have complementarity in terms of shape, in terms of chemistry, and then generate potential transformations, so potential set, potential complex. So stating that, let's say, this protein here will interact with human H-mark, this SARS-CoV protein, sorry, will interact with human H-marks via this interaction site. So the problem with the approach, they are extremely fast, but they typically don't work very well. Like they would provide reasonable results only in about a third of the case. And the reason is simply that it's too much of a rough approximation of the underlying physical process. We assume that the proteins are relatively rigid. They are not so flexible, so that you could look for a nice shape complementarity, but it's not always true. Sometimes also it's simply that this surface are quite flat, so you actually don't have this nice ideal case where you have a lot of convex and concave region which you can fit with one another. So this is what makes rigid body docking limited. Another mode of reasoning is what we call homology reasoning. Suppose that we know that two proteins, let's say the beige one and the purple one, are quite structurally similar. They look alike. And suppose that we have the same for protein 3 and 4. They look alike. Suppose, lastly, that we know for a fact that proteins 2 and 4 form a complex, and this will be the shape of this complex. Then we can reason based on this similarity and predict that proteins 1 and 3 will interact with one another, will form a complex, and the shape of the complex can be obtained by essentially superimposing the purple and the beige and cyan structures onto the structure of the complex. This is what we call called template-based modeling, and it is highly successful. The problem with template-based modeling is that by definition, this is not a very sensitive method. Suppose that you mutate you changed a few amino acids within the sequence of one of the protein. Well, template-based modeling will not be able to tell you whether the complex still exists or not. Unfortunately, we know in real life that a few mutations can break down a complex. This is the basis for viral escape. Why does an antibody work for one viral variant and not for another one? This is because sometimes a few mutations can completely break a complex, and homology reasoning cannot help you in this regard. So this is where another method to deal with, to reason, is also to do what we call local homology reasoning. The idea is the following. We believe that the function of proteins is often bound by local motifs, local regions on the protein. And this tends to be true. For instance, you can find certain patterns in structures that will be found repeated across many proteins and that can be associated with function. For instance, this is a so-called zinc finger. You don't really need to remember this name, but it tends to be associated with RNA binding proteins. So another way to perform homology is to look for these so-called motifs in proteins and reason based on this. So in this example here, you have two proteins that don't really look alike in terms of global shape, but they share common motifs, and hypothetically they would have they would perform a similar function. Okay, so then again, the question is, do we know enough motifs to predict the structure of the function of a completely new protein? This is not clear. And are these motifs sufficiently robust to, let's say, predict the effect of mutations? This is not clear either. So here comes machine learning, if I could say so. So how can machine learning help us? In principle, machine learning can help us learning this structure to function relationship. So from a given protein, you can extract so-called features that describe its shape. So for instance, for a given amino acid, which here is depicted in red, a few atoms, you can ask how exposed is it to the solvent? Is it more like on the exterior of the protein or more buried in the middle? This is a geometrical feature that you can calculate, and it would provide a description of what does the environment of your amino acid look like. You can compute descriptors that describe the curvature of the surface of the protein. You can have features that describe as well the so-called secondary structure motif, beta sheets, alpha helixes. Perhaps this is familiar, and so on. And you can compute a variety of such features based on physics, for instance, electrostatic potential, hydrophobicity indexes, as well as for evolutionary features. When I look at, let's say, other organisms, not just human, but let's say other primates, other mammals, is this amino acid so-called conserved, or does it intend to mutate? Using these so-called features, you can try to learn a function using machine learning. So let's say Suppose that I would like to identify for a given protein its binding sites, regions that bind with other proteins. In this example here, this would be the red amino acids of the blue protein here. So you can think of it as a binary label for every single amino acid of my protein, which is 1 if it's a binding site and 0 otherwise. And you would like to learn a mapping between these molecular descriptors and your target. So as you all know, machine learning is very appropriate to do this using supervised machine learning classifier like decision trees. And these methods would tend to work better and faster than traditional methods, especially when it comes to predicting the impact of, let's say, a single mutation or few mutations. These methods tend to be very sensitive. The only problem is that they are quite limited in accuracy in general, because these molecular descriptors are not expressive enough. They cannot capture all the wealth of the structural motifs that we are familiar about. Like, you know, from detailed studies of specific proteins, you can get to understand that some motifs are important, and you cannot really catch them using these handcrafted features. So this story is familiar to many of you probably. What's the answer to sort of limited expressivity is to do deep learning. Try to learn directly from data molecular descriptors that better capture the environment and the structure and the geometry of a protein. So this is a topic that has been of research topic that has been active for a couple of years now, but it's still very fresh. The main question is how do you actually represent these protein structures? Should you think of it as a point cloud? Like say 1 point for every atom, which is quite close to the actual structure file that when you sort of inspect them. And if yes, what kind of network would be suitable for this? Perhaps point cloud neural networks or other atoms. Another way to do it would be to think of it as like a density of atom and try to model it using 3D convolutional neural networks, so voxel-based representation. Here we have sort of the benefit that we know very well these architectures, 3D convolutional networks, but there is a problem of orientation. Protein structures are you expect their function to not depend at all on their orientation in space, right? It's arbitrary. Any orientation can be used to represent a protein structure. The problem with convolutional neural networks is that they are known to not be very robust to rotations. Another way to do it would be to use graph neural networks, try to represent the structure of a protein as a graph, with, let's say, each amino acid being a node and edges being, let's say, physical distances between these residues, or in these examples, binary edges representing Voronoi tessellation phases. This is actually a work that was developed here in Vilnius by Professor Venklovas and Dr. Oleshnovich. So this is another way to represent them. And you can also try to look at them as molecular surfaces and trying to analyze these objects. The point is that this is an open question, and this is where the tricky part is. So just in a couple of words, we have developed our own method for analyzing the structures, which is based on so-called geometric deep learning. The idea is to take a protein and look at its neighborhood, like local regions within the protein. You, let's say, take an atom, extract an atomic neighborhood, and orient it based on its covalent bonds, and then pass it through a set of what we call trainable spatiochemical filter that will learn an embedding, a representation of our neighborhood. So you can do the same at the amino acid scale level. And the general idea is that you will try to learn from raw data good molecular descriptors. And in a couple of words, we've been able to do it, and with some success on this task of protein binding site prediction. And for instance, you are able to recover, let's say, hydrogen bonds. You can show that the neural network can learn some chemistry directly from raw structure. In terms of performance, this is also quite exciting. You do significantly better than methods based on homology or based on handcrafted molecular descriptors. And just as an example to conclude, this is a prediction of the epitopes of the coronavirus spike protein. So regions that are likely to be targeted by antibodies. This is a heat map where the deep blue corresponds to amino acids that are predicted to be heavily targeted by antibodies, and white would be sort of neutral regions that are not targeted. And you can see that there's quite a nice match with what we know about how the immune system reacts about these proteins. So to conclude, I would skip this part and show you this graph. This is a depiction of a human protein-protein interactome. We have about 20,000 proteins in our body, and they are involved in about 650,000 interactions. Interestingly, it's not a lot. In principle, every protein can interact with every protein. The cell is an extremely crowded place, but 650,000 interactions is much less than the total number of interactions you could expect, which is basically 20,000 to the power 2. This means that these proteins are able to recognize each other very well, and we don't really know how. We don't know how do they recognize each other and what also the structural basis for function. So it's a very fundamental scientific question, and we hope that AI can help us address this question. There have been exciting developments in the field, new techniques for determining structures. I'm sure you have heard about AlphaFold, which is a method developed by DeepMind to predict the structure of a protein from its sequence. And also other teams have worked on geometric deep learning of protein structures in order to learn function of a protein from its structure. So it's a very exciting field. There's a lot to learn, and hopefully many interesting applications in terms of drug discovery as well as biomedical engineering. So with that, I would like to thank you for your attention, the organizers, in particular, Linus Petkavicius and Mantas Kazlauskas for inviting me, my collaborators, Dina Schneiman and Chaim Wolfson, as well as all my lab members and my founding organizations, Tel Aviv University, the Human Frontier Science Project, and the Edmond Safra Levy Center for Bioinformatics.
Is bigger? Yes, Jérôme, thank you so much for your presentation. On my schedule, it was a bit longer, but that's fine. I do have a few questions that I'd like to address to you. And since our conference is also in place to make the, let's say, difficult technologies more understandable for general audience, in a nutshell, how does your research in protein prediction can help human beings, for example, in solving health problems? Could you name a few cases, popularly?
Yes. Yes, absolutely. So if you understand, let's say, how proteins can recognize each other, you can understand, let's say, why is one genetic disease occurring? Why, when you mutate an amino acid, this interaction does not happen anymore and the patient gets sick. You can understand better how do viruses infect our body. These processes are mediated by protein-protein interactions, and you could try to learn how to block these interactions once you better understand them. So the main applications are drug discovery, trying to regulate how proteins work, both proteins from our own body and proteins from pathogens, and generically, bioengineering. Like, could we design proteins that can do things for us? Let's say, let's say, catalyze degradation of plastic, for instance.
Wow. Okay. Okay. And we also got a question from our audience that I would like to also address to you. Can these research also help in growing, for example, so much needed organs or fix some DNA issues that human beings actually experience?
Okay, it's an excellent question. For growing organs, not necessarily. There is the saying in complex systems that more is different. We can understand a single component, like a single cell, and not know how to grow organs. Like, it's almost a different topic in the sense of research. You need to understand how proteins would like cells would self-organize, and it's like a different scale, if you wish.
Okay. Okay, so then thank you so much. Thank you so much for your presentation. Thank you so much for exploring such a, and in some cases, it seems like a difficult topic. But thank you once again for making it much easier for us to understand. Ladies and gentlemen, that was Jérôme Tubiana, postdoctoral fellow at the Tel Aviv University. And now we're moving to a short networking break, and stay tuned for more keynote speech in less than half an hour.